Hi, thanks for coming. My name is Manuela Ramalho. I'm a postdoctoral researcher at Cornell University working with Dr. Leslie Babonis in the Babonis lab. And today we will talk about unraveling the role of nanos in somatic cell differentiation in the Nematostella vectensis, a sea anemone. So before starting, I want to give a little bit of background. So what is nanos after all? So nanos, it's a zinc finger protein that is likely function as a transcriptional and translational repressor. Was originally isolated in Drosophila, plays an important role as anterior and posterior patterning, as you can see by this image here comparing a Drosophila wild type with a nanos mutant. This role of nanos was also found in other insects. This role for anterior and posterior pattern was also found in other insects. As you can see here, one example in ants. But nano was found in other animals too, and different than in other insects. Nano seems that there is no evidence in this other non-insects animal that they are working as an anterior posterior pattern, just in insects. So what is functions of nanos in non-insects animals after all? So turn out that nanos has two functions in non-insects animals. So for one of the functions seems that they are work as a germline, which means that they are able to work as a different ways in order to produce a viable gametes in its driving germ cell development. But they also can work as somatic cells. In this case, it specifies stem cell identity. And when we look at the distributions of nanos in a phylogenetic tree of animals, you see that all the animals has nanos. And you also can see that the most conserved function is the germline function, as you can see here highlighted for the blue star. But you can also see that some of these groups of animals also are known to have nanos providing somatic functions, and this is less studied. Interestingly, it appears that the most of these animals also have two copies of this gene, and one copy seems to be involved to a germline, and the other seems to providing a somatic function, suggesting that this gene has duplicated and give a rise to a new function. So that's why, in general, we are interested to understand how the duplicated genes acquire new functions. And as the germline functions, they're much more studied, conserved, and consistent across all the animals, lineage of animals, to understand the new functions of how these duplicated genes acquire new functions, it makes more sense focus on the somatic functions. So how these new nanosomatic functions arise. So that's why we are pursuing in this talk today. And which brings us for the title of our talk that we started today, and unraveling the role of nanos in somatic cell differentiations in the sea anemone Nematostella vectensis. So before we move further, I want to return for our original phylogenetic tree of animals, and I want to expand this Cnidaris branch in two other branches. So one is the Medusozoa, that is here represented by the hydrozoans and the jellyfish, and the Anthozoa that here I'm giving an example of the sea anemones and the corals. So previous study was able to study nanos in Medusozoans, and in those studies, they were able to demonstrate that nanos specify stem cells in Clintia and Hydroactinium. So if you look with more details here in this image, you can see that nanos, it's highlighted in purple, and they are identified in related to development of stem cells. In the Nematostella, nanos was already studied before by Extavor and collaborators in 2005, and they were able to study, to demonstrate that in sea anemone, Nematostella vectensis, they have two copies. that we will call in this talk today the somatic nanos and the germline nanos. So the germline nanos seems to be also related to the germline functions in the Nematostella vectensis, and the other gene, the somatic nanos, this author, they suggest they are also related to regulated stem cells development. So when we look at the pattern for expression of these genes in Nematostellas, when we look at the gastrulate stage, the germline nanos, they seem to be expressed in the germline. So it is expressed in this image here in the endoderm, which will give a rise to the germline. And when we look at the same stage, but now for the somatic nanos, so it's a gastrulate stage, but somatic nanos now, you can see that it is expressed in somatic cells. So nanos is expressed here in a salt and pepper pattern in the ectoderm. So just by looking the different way of they are expressed in this gastrulate stage, you can see that they have a different functions. And just a quick reminder, we are focusing the somatic nanos in our talk today. So our hypothesis for this paper, as the same way in another cnidarians, we believe the somatic nanos in the Nematostella vectensis, this regulated the stem cells development. And in order to pursue that, let's first look how we produce stem cells. So previous studies here was able to demonstrate the pathway in order to produce stem cells. So we started for the SOX-B2 gene, and then we have two different paths. One is for producing neurons, and the other is for producing stem cells. So we know that zinc finger, PAX, and minicollagen, this is all genes in the pathway to produce stem cells. So we will approach our questions in the back order. So we will first focus in the PAX genes and minicollagen. So when we focus in the larvae now in the somatic nanos and the PAX-A, we are focusing, PAX-A is a stem cell transcription factor. And when we look at the PAX-A, we are seeing that somatic nano is not co-expressed with PAX-A. And when we focus now in the minicollagen, you can see that minicollagen is a structural protein that is specified also for stem cells, and they're also not co-expressed with the somatic of nanos. So now we move one step behind in our pathway, and now we are looking in the zinc finger. So knockdown of zinc finger, that is also a stem cells specified regulatory gene, they don't have any effect in somatic nano expression, as you can see by this image. There's no difference between the white type and the knockdown zinc finger. So nanos, it is expressed normally here. So now let's go further. Let's back one step behind. For now, focus in the SOX-B2. Okay, so when we knock down the SOX-B2, we do saw that there is effect in the somatic nanos. So, which brings us the somatic nanos, it is expressed downstreams of SOX-B2. Okay, so now, based what we just saw by these results, we have a new hypothesis that is somatic nanos, it is involved in the neuron development, as you can see by this here. So, which means that somatic nanos is probably here in this pathway to produce a neuron. In order to pursue that new hypothesis now, we knock out the somatic nanos. So how we did that? So we inject the CRISPR-Cas9 in the zygote, and we grow, that it's a guide RNA. So we grow our adults of these animals, and then we cross them. And then we grow this offspring of F1, and we expect that we'll be doing these experiments now in the larvae. But spoiler alert, we could not do that because our animals, they die. So knockout, the somatic nanos cause the embryonic inviability. So when you compare the percent of survival, as you can see in this graph, you can see that our nanos, mutant animals, knockout, they are not able to go further than cleavage. So we are not seeing even like evidence for fertilizations. And when you compare with the wild type, you can see that there's a huge difference. So we cannot get our animals growing in order to test if they're involved in the neurodevelopment. And another interesting phenotype that we also saw, so the knockout of somatic nanos reduced the spawning activity. So we started in this graph here, looking for the total of spawn animals in percentage, like starting from 100%. And we start to see a huge decrease of animals that it's able to spawn, as you can see by this graph. And even if this animal seems to be recovering and start to produce egg again, gametes, again, these gametes are not viable anymore. So the conclusion that we have for this talk so far, it is the somatic nano has the germline and the somatic functions in Nematostella vectensis. So we started with this original hypothesis, thinking that one gene in Nematostella will be responsible for the germline function and the other gene responsible for the somatic function. But there now that in Nematostella, it seems that this somatic function has these two other functions, the germline and the somatic function. And compared with other cnidarians, we are also seeing that these somatic nanos, they are not producing, they have a different function than other cnidarians. So in hydroactinia, the somatic of nanos seems to be related to producing stem cells, and in Nematostella could be related to neurons, but we already have evidence that it's not related to stem cells. And to really test the hypothesis if the somatic nanos is related to produce neurons in Nematostella, we now need to knock down the somatic nanos in order to have viable animals to see if they're able to work in the development of neurons in Nematostella. And the future directions, we also want to inject Nanos RNA into our zygotes of Nanos knockout in order to try to rescue the normal development of these animals. So with that, I would love to say thank you for all of you. And if you have any questions now or in the future, please contact us. I also need to acknowledge Cornell, my house now, and NSF for providing fundings. All my friends and from Babonis lab, and this is our contact. So thank you so much.